This is PediaCast.
Welcome to PediaCast, a pediatric podcast for parents. And now, direct from the campus of Nationwide Children's, here is your host, Dr. Mike.
Hello, everyone, and welcome once again to PediaCast. It is a pediatric podcast for moms and dads. This is Dr. Mike coming to you from the campus of Nationwide Children's Hospital. We're in Columbus, Ohio. It's episode 517 from May 5th, 2022. We're calling this one Natural Killer Cells and Childhood Cancer. I want to welcome all of you to the program. So we have a really interesting episode for you this week, especially if you enjoy the science side of our conversations, which I know many of you do, because one of our most popular episodes at the height of the COVID-19 pandemic was one called How Our Immune System Works. And if you missed that one, I will put a link to it in the show notes for this episode, 517, over at pediacast.org, so you can find it easily. Today's episode sort of builds off that one as we consider a particular type of cell in the immune system, one called natural killer cells, also known as NK cells, because, these cells don't really like to be referred to as killers. So scientists made a softer, less controversial name for them, NK cells. However, they really are killers, and they kill other cells in the body, and in particular, they kill cells infected with viruses because, remember, viruses hijack cells and turn them into virus-producing machines or factories. So natural killer cells destroy the factory by killing the cell. Well, it turns out NK cells also kill cancer cells, which is pretty cool because that provides opportunities for isolating these cells, growing more of them in the lab, genetically modifying them to be more efficient and effective at tracking down cancer cells, and killing them while leaving non-cancer cells alone, which is something that chemotherapy and radiation is unable to do. So there's still much work to be done, but we are definitely making progress with these natural killer cells, or NK cells, especially in the treatment of cancer. And our guest today is an expert on NK cells, Dr. Dean Lee. He is a principal investigator with the Center for Childhood Cancer and Blood Diseases at Nationwide Children's Hospital. And his lab has developed techniques for working with these natural killer cells with the aim of using them to fight childhood cancer. So we'll have an interesting and science-y conversation today, yes, but we will also break down the terminology and explain things as we go. Dr. Dean Lee will be here soon, but first let's cover our usual quick reminders. Don't forget you can find PediaCast wherever podcasts are found. We're in the Apple and Google podcast apps, iHeartRadio, Spotify, SoundCloud, Amazon Music, and most other podcast apps for iOS and Android, including a new one, Good Pods. We are now in the Good Pods app. And if you like what you hear, please remember to subscribe or follow our show so you don't miss an episode. Also, please consider leaving a review wherever you listen to podcasts so that others who come along looking for evidence-based child health and parenting information will know what to expect. We're also on social media. We love connecting with you there. You'll find us on Facebook, Twitter, LinkedIn, and Instagram. Simply search for PediaCast. And we have that handy contact page if you would like to suggest a future topic for the program. You'll find that at pediacast.org. I want to remind you the information presented in every episode of our program is for general educational purposes only. We do not diagnose medical conditions or formulate treatment plans for specific individuals. If you have a concern about your child's health, be sure to call your healthcare provider. Also, your use of this audio program is subject to the PediaCast Terms of Use Agreement, which you can find at pediacast.org. So let's take a quick break. We'll get Dr. Dean Lee settled into the studio, and then we will be back to talk about natural killer cells. It's coming up right after this. Dr. Dean Lee is a principal investigator with the Center for Childhood Cancer and Blood Diseases at the Abigail Wexner Research Institute at Nationwide Children's Hospital. He's also a professor of pediatrics at The Ohio State University College of Medicine. Dr. Lee has a special interest in our body's natural killer cells, which are an important part of our immune system's response to viral infections and cancers. Turns out natural killer cells can be genetically engineered in the lab to respond more effectively to some cancer cells, which holds promise as a treatment for cancer, one that is highly selective of the cells we want to eliminate. More specifics on the science of that in a moment. But first, let's meet our guest, Dr. Dean Lee. Thank you so much for stopping by the studio today.
Thanks, Mike. Glad to be here.
Yeah, I really appreciate you taking time out of your busy schedule to join us. I think a good place to start would just be a definition. What exactly are natural killer or NK cells?
So we have a variety of kinds of white blood cells in our blood, and those have all kinds of different functions. You can broadly split them into cells that are of what we call myeloid lineage, the neutrophils and monocytes, and cells that are lymphoid lineage. In the lymphoid category, there's B cells and T cells that lots of people know about. Those are the cells that respond when you get a vaccine. And then there's NK cells. And NK cells are called that, very creative name calling, right? Because they had this natural ability to kill cancer, just when you look at them in a dish. So B cells and T cells have to be trained. You get a vaccine, you train those cells to recognize chickenpox, and for the rest of their lives, the B cells and the T cells that responded will only recognize chickenpox. NK cells don't really look at specific things. They look at general signs of safety and danger, and therefore look at naturally occurring pieces that tell them that a cell is dangerous and will kill those cells without ever having been trained to do so.
Yeah. And that would include cells that are making virus, because when we get sick with a virus, our cells turn into little factories making more virus. So
Right.
the NK cells will attack those cells and kill them to stop the factory. But then they can also recognize there's something different about this cell that is a cancer cell and attack those as well.
In both cases, the cells are stressed. So the virus puts a lot of stress on a cell to be manufacturing all those virus particles. So in terms of metabolism, and sometimes even in terms of DNA damage, and in terms of internal recognition of these foreign nucleic acids and foreign proteins. So the stress then causes what we call stress receptors to come up on the surface, and an NK cell sees those stress receptors and says, there's something wrong with you. You're not behaving normally. Cancer does a similar kind of stress. It induces defects in the DNA. There's lots of mechanisms for not repairing DNA in cancer, and metabolic stress, because cancer cells are just really growing fast and putting that kind of stress on the cell. And that induces the same kind of stress receptors, stress ligands on the surface that the NK cells can see.
Yeah. So these are really the first cells that often respond to problems, because as you mentioned, the B cells and the T cells really have to, so the T cells have to make more of themselves in order to respond to a certain invader, like a virus or a cancer. And the B cells have to start making antibodies, and there's sort of a delay in that process. So those NK cells really are the first line of defense of the immune system, right?
Mm-hmm. Yeah, important for bridging the time that it's going to take for the T cells and B cells to respond, and also for setting up the kinds of signals that tell the B cells and T cells that something needs to be responded to. So they're sort of like the first wave of the Green Berets. Not only do they clear out space for the rest of the troops to come in, but they also send out the signals that everything's ready and this is where we need to go.
Yeah. During the COVID-19 pandemic, people heard new words like cytokines. Is that the chemicals that are being used as signals to recruit other immune system cells to come in and help with the fight?
Right. So a variety of cytokines that are what we call pro-inflammatory, or set up those signals that say this is the right place to have an inflammatory response.
Yeah. I just want to point people to a podcast that we did sort of at the height of the COVID-19 pandemic called How Our Immune System Works. It was PediaCast episode 462. And I'll put a link to that in the show notes for this episode 517 over at pediacast.org, because it really gave a big overview, and natural killer cells were a part of that talk. And now we're going to really focus in on those particular cells. As we think about cancer, how is it that the NK cells then respond, so that they've attached to the cancer cell, they send out signals? What else do they do?
Cytokines are proteins that are sort of loose in the blood to create signals that can travel. And they can either signal other cells to come and sort of attract those other cells to the area, or they can activate responses that are in local cells, or they can suppress responses. So we have some cytokines that actually calm things down, some cytokines that activate things. So NK cells produce a little bit of both, depending on the circumstances. And the primary cytokines that they release are ones that are going to attract the rest of the immune system to come in. But they also have some cytokines that have killing ability themselves. There are receptors on virus-infected cells and cancer cells that are what we call death receptors. And when NK cells make those cytokines, they can travel around and trigger any of those cells that have those receptors open. The second arm of what NK cells do are to specifically release granules of toxins to a cell to intentionally kill one cell. So rather than traveling around and interacting with any cells that have those receptors, this is a targeted release of almost like little payloads to the cancer cell or virus-infected cell. And those contain mostly two proteins. One is called perforin. Again, a really creative name because it perforates a cell, so it pops a hole in the cell. And then granzyme that goes in through that hole and initiates a whole cascade of events for cell death. So the perforin and granzyme side doesn't need a receptor. It doesn't need something on the cell that says it's going to be sensitive, but the cytokine part does.
Yeah. And when we heard about those cytokine storms that maybe have been related to severe COVID, that's when you have a lot of those, the cytokines that cause the immune system to react and up the immune system, and not as many of the ones that sort of calm things down, right?
Right.
Yeah. Now, in a perfect world, then, this would just kill every cancer cell in our body, but obviously that doesn't always work out. So cancer cells do have some means to avoid the effects of NK cells, right?
Mm-hmm. So the natural way that NK cells see those, like I mentioned before, is those stress ligands that get recognized by the receptors on the NK cell. And cancers can learn to shut those stress receptors down. And over time build up enough of the suppressive cytokines that kind of make the NK cells a little bit dysfunctional. So once they're in a tumor, they can't function as well. For the first part, as far as helping the NK cells recognize better, it goes back to your comment at the beginning about genetically modifying them. So there are ways that we can enhance how an NK cell can see a specific target, either with an antibody or with this genetic engineering. On the suppressive side, there are ways that we can make the NK cells feel better in those suppressive environments and retain their function and not be as sensitive to that suppression.
Yeah. I just want to point out that this is inside of our body, sort of a natural selection process, right? Mm-hmm. So if you have, cancer cells aren't really smart enough to say, oh, I need to start making something that's going to make the NK cells not work. It's just that the ones that happen to start doing that, then the NK cells aren't going to be as effective. And so those particular cells are the ones that are going to multiply and become kind of resistant. And it's sort of the same thing when we talk about natural selection on an organism level. It's the same kind of thing happening. It's just inside our body.
Mm-hmm. So it turns out we actually probably develop cancer a lot more than we think we do, but our body takes care of it. And occasionally you get one that has somehow mutated in just the right way to escape that. And so finding ways to overcome that escape then is sort of the next wave of what we need to do with those cells.
Yeah. And so basically, once you genetically engineer those cells to overcome any resistance that a cancer cell might have, then you can infuse those in the body, and those NK cells can go take care of the cancer.
Right.
But the problem is getting enough of those NK cells, right? Tell us about that problem.
So we've known about NK cells since the 1970s, but our knowledge about them compared to other lymphocytes like the T cells and B cells is probably 15 to 20 years behind. And in part because we didn't have a good way to grow them. So for T cells, we've known 4 or 5 different ways to grow them since late 70s, early 80s. And B cells, the whole idea of monoclonal antibodies was that we learned a way to immortalize B cells and make them live in a culture dish for decades. And that was in the 1980s. But NK cells were tough. They just didn't seem to grow in response to the same kinds of signals. And if you can't grow the cells in the lab, you can't study them very well. And if you can't study them, you don't know how they work. And if you don't know how they work, then nobody cares about them. So they kind of smolder along, with a few people interested here and there. In mid 2000s, I was working on T cells, and we had generated some feeder cells to help the T cells grow. And so this was sort of our process for growing more T cells. And every once in a while, those cultures would get overgrown with NK cells. So my mentor at the time, he said, well, here's a couple of ideas. Why don't you play around with it and see if you can actually more consistently get them to grow and intentionally grow the NK cell part? It's like, nobody cares about NK cells. I'm a T cell guy, But I went ahead and did it. And sure enough, we found kind of the secret sauce for getting these cells to grow. And now on a very routine basis, we can get 2,000 to 3,000 times as many in a couple weeks, 30,000 times in 3 weeks. So we have lots and lots of cells that we can generate in the lab, not just to study, but now to think about giving them to patients. Because if you can't grow them to large enough numbers, they can't be effective.
Yeah.
So it's really helped us on both sides, both on the studying side and on the therapy side.
Yeah. Before you discovered this, when you wouldn't really have enough NK cells to infuse into a patient, but by the time that you had enough, their cancer would be really be more likely to be progressed and invasive and all of those things, because you need a lot of them quickly.
The original studies that were done to deliver NK cells would take a family member and put them on a machine called an apheresis machine, where we would basically take all of their blood to cycle through the machine, separate out the white blood cells, give back all the platelets and plasma and red cells. And you'd be on that machine for 4 to 5 hours and give up as many of those white blood cells from your body as you could. We'd take that and get rid of the T cells, because they actually cause some problems if you go between people. Put the rest of the culture in some cytokines that NK cells like to activate them overnight. And then that's what we would give to a patient as an NK cell therapy. And it was a one-time shot. Like you couldn't keep doing this to the donor. So, and doing that, you could give somewhere around 20 million NK cells for every kilo of body weight of the patient. So it worked. It worked a little bit. We had some hints that they were doing some benefit, but that was it. We couldn't get beyond those numbers. Now, instead of getting 20 million cells per kilo, we can easily give 100 million, even more than that. We can give it multiple times. So we have several clinical trials where we've given 6 doses of 100 million cells every time. So we're at, 10 to 50 times as many NK cells as what we could give before.
Yeah. Now, are these, so when you say the T cells could create a problem, we think about blood types, and that there's a problem with rejection when there's organ transplants. And since these are cells, are they coming from that person or just someone who has a similar enough cell that won't be a problem? Or is that not an issue with NK cells at all?
With NK cells, it's not an issue. For T cells, it's the way they see the world. And so sometimes I explain this a little bit like passports, that T cells are really trained to see a very specific person inside a specific passport. And that's how they're trained. And so if you took that same picture and you put it in a different passport, they wouldn't recognize the person anymore, because they go, oh, well, that's somebody who looks similar, but they're from England. They're not from the United States. And so they would sort of ignore them. And the passport that I'm talking about is this protein called MHC, and it's present on all of our body cells. And its job is to show the immune system what's inside. And as long as it's just showing, yep, I'm just the normal pancreatic cell that you've always seen, there's no problem. The moment it shows that passport with a virus inside, that's when the T cell can see it. NK cells don't look at the picture. They're only looking to see that you have the passport. They just want to know that you actually are showing the rest of the immune system who you are and that you had sort of valid documentation. So there's a variety of these that NK cells see, and the way that they see them from one person to another is mostly on the being inhibited side. So they don't overreact to wrong passports. They just go, oh, you don't have the right one.
Yeah. And so those proteins on the surface are sort of different for each person, and specific to someone, and your own immune system is trained to recognize that as, hey, this is you. A normal cell don't attack this one. And so the NK cells are going to be able to do that in anybody. So in other words, you could just have an off-the-shelf product with NK cells, and it doesn't really matter who they're from, and they're going to help as long as they're trained to fight whatever it is that you want them to fight.
That's our goal. So far, all the studies we've done have been, like I said, with family members, initially by that apheresis process, and then secondly by expanding them, like I mentioned. But now that we understand better how well we can expand them and what we think are kind of ideal genetic characteristics for NK cells, we're working on another approach to really look through the entire country and use the same group that does bone marrow matching, the NMDP, has worked with us for the last 2 years to identify people who have the right kind of MHC molecules and KIR to make ideal NK cells. And we're harvesting large amounts of NK cells from them, splitting them up into banks. And from one individual, we can now make enough NK cells to treat 20, 50, or even 100 people.
Yeah. Wow. That is amazing. So what do you think in terms of how far off are we from having a product like that? I mean, I know you can't predict the future, but I mean, are we talking like a decade or shorter than that?
Well, we've treated our first 6 patients over at OSU with this off-the-shelf universal donor product. We have our first trials open here at Nationwide to treat kids. I think we'll treat our first ones in the next couple of months. And then it will just be a matter of seeing whether they actually do work the way that we intended. If it works out that they're as effective as we hoped, then the next path is really to find, can they be made efficiently by a company, distributed, delivered at multiple hospitals, and can we expand the delivery mechanism for those?
Yeah. Now, we talked about genetically modifying these cells in order to make them more efficient and effective at a particular cancer. How do you go about genetically modifying a cell? And I will point out that we did an episode of PediaCast CME, which is our continuing medical education podcast. But folks we'll put a link to this one in the show notes as well. It was Genomics 101, an introduction to next generation sequencing. And we did try to explain things as we went along, because even primary care docs like myself, this is all still very foreign. We learned about it in medical school like a long time ago and then forgot about it. So I think we did a pretty good job of explaining as we went. So folks who are interested in genomics and genetic sequencing and sort of how cells could potentially be modified, that would definitely be a good episode to listen to if you're interested in that. But for your purposes, you're really just swapping out DNA, right? Because that's a code for what proteins get made and get shown on the surface.
Yeah. So for the NK cells, there's 2 things we'd like to do. There are some genes we'd like to get rid of, the ones that kind of calm the NK cells down. We want to make them not sensitive to those things inside the tumor. And the other is to put in new receptors that allow them to specifically target the tumor or give them new function. Both of them are similar approaches. They're both considered genetic engineering, whether you're genetically engineering away from a gene or towards a new gene. But most of our experience in medicine has been with adding new genes. And so we started with that maybe 15, 18 years ago, because we have lots of experience of engineering T cells. So CAR T cells that we use now as a commercial product to cure leukemia in our kids, is used by adding one of those genes to a T cell. Unfortunately, it's a good thing, I guess, from biology perspective, but because NK cells are the first responders to viruses, they have very high turned on mechanisms for sensing viruses, and they would rather die than be infected. So we did a whole bunch of work trying to get the same viruses that would help us engineer a T cell to work for an NK cell, and none of it would work. In the last 4 years, we've had a big growth of things that we're doing in the lab, and there's a new approach called CRISPR that Mesam Kararudi in my lab was able to adopt and learn how to get rid of a gene. So rather than putting in a virus, you can put in little small pieces of RNA and protein that target a particular gene and able to make cuts in it. And that the NK cells tolerate fine. And then if you follow that with a new piece of DNA that has the right signals on the ends to insert, you can put in that new gene at the same place that you just cut out the old one.
Yeah.
And that's actually working really well. We're getting very nice 80 to 90% efficiency. It's a very careful, controlled editing, unlike some of the other viruses where you don't really know where it went. Here, you actually can tell the gene exactly where you want it to be in the genome.
And how do you figure out even what gene to add? So do you just look at NK cells that you know work and then look at their genome to see how they're different than maybe one that doesn't work and compare, and that's how you figure it out? Yeah, I'm just using the knowledge that I learned in that Genomics 101 episode to try to wrap my brain around how do you know what gene to put in to begin with?
So the genes we're putting in are mostly ones that we've had about 20 years of experience building. Lots of labs around the world are looking at these genes called CARs, Chimeric Antigen Receptors. And they're pretty consistent structures that are an outside piece that's from an antibody that tells you what to target, and an inside piece that says what to signal. So if we understand enough about NK cells to know what a good signal is to an NK cell, we can put those pieces on the inside so they get good signals. And if we understand the tumor well enough to know what kinds of things mark that tumor as being tumor and what we can target on them, then we can put that on the outside of the protein. And so that's really all a CAR is trying to tell a NK cell, if you see this marker, kill that cell because that's a bad cell. For the genes to take out, some of it has been by luck, just finding them. Some of them have been genes that we know are a problem. Some of the suppressive pathways we've known about for 20 or 30 years. And if you can get rid of the receptor to that, then you can eliminate the ability of the tumor to suppress the NK cells. So we have pretty targeted ones that we can look for.
Yeah. And since they are so targeted and to a specific cancer, what cancers right now have this therapy available?
So the only one that's been done in a clinical trial and published is CD19, which is the same thing we target CAR T cells for leukemia. Also useful for lymphomas and some other blood cancers. There are dozens of clinical trials that are available for solid tumors. None of them have had the kind of dramatic effect that the CAR T cells had for leukemia. And some of that is because of the different ways that T cells and NK cells behave. So our first move into solid tumors with a CAR will be to put the CAR into the NK cell. And there's a couple of already pretty well-established targets to try that with. One is called HER2. It's the same target that the breast cancer drug Herceptin is made for. There's another one called GD2, and that's the target for an antibody we use in pediatrics for neuroblastoma, dinutuximab. So we already have a little bit of experience with targeting those things with antibodies, and it's not such a big leap then to tell the FDA we want to target that same thing with an NK cell.
Very interesting. Now, we still have chemotherapy and radiation, which are not specific to the cancer cells. It's just going to kill or destroy any rapidly dividing cells in your body. And so it's going to just, be harmful to normal cells as well, whereas the NK ones are going to be very specific for the cancer cells. But we're not at the point where you can only use NK cells, correct?
Correct. So far, cell therapy has been remarkable in some settings, like the leukemia that we mentioned, where essentially a single infusion of CAR T cells induced a long-term cure for many patients, but not all by themselves. We still need to treat patients with a little bit of chemotherapy to get the patient ready for those cells. And there are some other therapies that sometimes we need to add afterwards, either to reduce some toxicity or to help the cells to move along. At some point, we may be able to do enough engineering of the cells that you don't need that initial chemotherapy, but I don't entirely know that that's the right approach. I think there's some equipoise around how we think of what genetic engineering we're doing, and we might be moving towards or a little bit over engineering, right? It's because there's small amounts of chemotherapy that are really very easy, very inexpensive to give, very well tolerated, that we shouldn't kind of throw out the baby with the bathwater, so to speak.
That makes sense. I mean, ultimately, we want good outcomes.
Right.
And so you got to figure out what the right combination of things is for the outcome that you want. As people, and you talk about over-engineering, and I think in a lot of people's minds, could be kind of a scary thing, thinking about taking someone else's blood, genetically making the cells different, and then infusing them into another person. So are there any dangers or risks involved with that process?
There are. Anytime you're making changes to the DNA, there's a possibility for changes that you aren't aware of. I think doing dangerous things to the DNA intentionally is much less likely. I mean, we know what we're targeting, and both our own regulatory structures within the hospital as well as the FDA are very strict about what kinds of proof you have to give them, that you're making safe changes. But it's the unknown ones. And there have been a few, very few, but some cases where, for instance, we were genetically engineering bone marrow to try to cure someone of a immune deficiency, and instead the engineering triggered a gene that caused leukemia. So those kinds of things are very, very carefully looked at. The FDA wants lots of data ahead of time to make sure that you know what you're engineering, that it's not close to any of these other dangerous genes, and that you've done due diligence to make sure that you've got a product that is as safe as you can make it.
Yeah.
Ultimately, we're treating patients with cancer. So there is a sort of accepted amount of risk that that patients themselves are willing to take, knowing that they've got a bad disease and are wanting to try new cures.
Yeah. I mean, for all of this, and really for any decision in our life, we think about risks versus the benefits and then make a decision based on that. I mean, even when we get in a car, the benefit of getting somewhere fast and, the risk of an accident, you're still weighing the risk and benefit. And so with these patients and families, I'm sure you go down that road of this is, the risk of not doing it is that this cancer may not be treated with standard therapy and could be deadly. But the benefit of using this, you would want that to outweigh the risk of using it, right?
Right.
And kind of walking the family through that decision-making process and making the decision together.
Mm-hmm. And we have a long history of doing that for all of medicine, but very specifically in cancer therapy. We know that chemotherapy and radiation is toxic and hurts people. And so we do as much as we can to effect good cures. And then we do as much as we can to back off from that and do as little damage as possible. Yeah. And so it's always a little bit of that trade-off. You're trying to do as much good as you can with as little bad as you can.
Yeah. So what does the future hold for manufactured immune system products? Where do we go from here?
So you mentioned one of them, and that's an off-the-shelf product. I think we're at the very beginnings of that for NK cells. There's a lot of work being done about doing that for T cells as well. They're a little bit harder to figure out all the right pieces that have to be done to make them transferable from one patient to another without problems, but some good progress on that. So having something that is just readily available on the shelf that you can thaw and infuse is one of the biggest sort of forward-looking pieces. And that's because to do this kind of patient-specific manufacturing and engineering is not only expensive, but it also takes time. And many of these patients don't have the time to wait. So we have quite a lot of cases where we have patients who are, wanting to get one of these new therapies, but the amount of time that it takes for us to make it is longer than they can wait for their disease. So it is both trying to reduce the cost as well as, capture those patients that otherwise wouldn't be able to get therapy.
Yeah. In terms of insurance and coverage and for getting these kind of products, is that something you have to fight with insurance companies, or because they're clinical trials at this point, their medicine is provided free of charge, or how does that all work with the family? Because I'm sure that's probably a stress and strain on folks too.
For the ones that have been approved, like the CAR T cells, the insurance companies have actually been really good about covering them. They are very effective and completely effective, and have a really low number of toxicities to deal with, so their safety profile is high. And I think that everybody understands that even though they're expensive, so is not curing cancer, and so is curing cancer poorly. So that part has not been very difficult. For the ones that are currently in research, those are almost uniformly paid for by the researchers or by the hospitals. We don't feel that it's that it's ethically appropriate to charge somebody for something that's experimental. And so we build in as many possible ways of covering that as possible. And we get government grants, we get foundation grants, philanthropy that comes to the hospital. And in some cases, when it's part of an overall treatment package, we try to bundle that in with the other things that are standard, just so that we can help move along better standard of care.
Yeah. So when folks donate money to a children's hospital, this is the kind of thing that money goes toward helping provide this type of research and giving those medicines to children who need them.
Absolutely.
Yeah. Very, very important. I know you have a lab at Nationwide Children's, the Lee Lab. Tell us a little bit about that.
So all of it really started short after my training in building T-cell therapies. And as I mentioned, we pretty quickly switched to NK cells, mostly on a fluke of finally figuring out how to grow them. When I moved here to Columbus, I only had one person who stayed with me who was a graduate student. So we had to kind of rebuild all those people. And now we're up to 10 or 12. There's a wide variety of fully trained staff scientists who have their PhD and finished postgraduate training. We have PhD students. There are technicians, some of which are pretty freshly out of college. Fellows who are in their clinical training who want to come through and understand better about what a research lab does before they go back to the clinic, and even a couple of veterinarians. So.
Great. And we'll put a link to the Lee Lab at the Center for Childhood Cancer and Blood Diseases at Nationwide Children's Hospital so people can learn more about what you do. There's also a great article in the magazine Pediatrics Nationwide called Optimizing the Body's Natural Cancer Killers. And that's a great article. I'll put a link to that in the show notes as well. And then tell us about the Center for Childhood Cancer and Blood Diseases. We've mentioned that a few times. What is that center all about?
It's about childhood cancer and blood diseases. In our clinical practice, we divide people into specialties. You have rheumatologists who study autoimmune diseases and doctors of infectious disease that study infections, and your cancer doctors and hematologists that study blood diseases and cancers. So on the research side, I think it's very similar, right? We want to structure the kinds of research that we're attracting into groups of people who can work together and collaborate. And in many places, individual research labs like mine would really just be kind of on an isolated island. We study NK cells, and we study NK cell biology because we want to know how they work and we want to engineer them better to cure cancer. But it's actually much more productive for me to have a lab next to me that also studies the biology of osteosarcoma, for instance. It's a cancer that I want to treat. And if I only know how the NK cells work and I don't have somebody who is thinking about how the cancer works, then we miss out on half of the biology that's happening inside of a person. So the CCBD is really structured to try to enable those kinds of interactions, whether you are looking at drugs, chemotherapy, radiation, the immune system, genetics. That might be the science that you're doing, but we're all studying different aspects of the cancer itself. Yeah.
And collaborating amongst yourselves and then at other centers, at other children's hospitals, and really sharing information and advancing the field together.
Right.
Yeah. Well, we really appreciate you stopping by and educating us about NK cells. Lots of links in the show notes this week, some of them that I've already mentioned, that Genomics 101, an introduction to next generation sequencing, PediaCast CME, and then the one, How Our Immune System Works. So if you're interested in genetics and genomics, and the immune system, please check out those episodes. And again, we'll put links in the show notes for you. So once again, Dr. Dean Lee, Principal Investigator with the Center for Childhood Cancer and Blood Diseases at Nationwide Children's Hospital. Thank you so much for stopping by today.
Thanks, Mike.
We are back with just enough time to say thanks once again to all of you for taking time out of your day and making PediaCast a part of it. Really do appreciate that. Also, thanks to our guest this week, Dr. Dean Lee, Principal Investigator with the Center for Childhood Cancer and Blood Diseases at Nationwide Children's Hospital. Don't forget, you can find us wherever podcasts are found. We're in the Apple and Google podcast apps, iHeartRadio, Spotify, SoundCloud, Amazon Music, and most other podcast apps for iOS and Android, including that new one, Good Pods. So be sure to check that one out. Also, our landing site is pediacast.org. If you go there, you'll find our entire archive of past programs, along with show notes, our terms of use agreement, and that contact page if you would like to suggest a future topic for the program. Reviews are helpful wherever you get your podcasts. We always appreciate when you share your thoughts about the show. And we love connecting with you on social media. You'll find us on Facebook, Twitter, LinkedIn, and Instagram. Simply search for PediaCast. Also, don't forget about our sibling podcast, PediaCast CME. That stands for Continuing Medical Education. It is similar to this program. We continue to turn the science up a couple notches, and we do offer free continuing medical education credit for those who listen, including not only doctors, but also nurse practitioners, physician assistants, nurses, pharmacists, psychologists, social workers, and dentists. And since Nationwide Children's is jointly accredited by many professional organizations, it's likely we offer the exact credits you need to fulfill your state's continuing medical education requirements. Of course, you want to be sure the content of the episode matches your scope of practice. Shows and details are available at the landing site for that program, pediacastcme.org. You can also listen wherever podcasts are found. Simply search for PediaCast CME. Thanks again for stopping by, and until next time, this is Dr. Mike saying stay safe, stay healthy, and stay involved with your kids. So long, everybody.
This program is a production of Nationwide Children's. Thanks for listening. We'll see you next time on PediaCast.